Histology plays a vital role in understanding the microscopic structure and composition of tissues and organs in the human body. The study of tissues is facilitated by the use of various staining techniques, with hematoxylin and eosin, H&E, being the most widely employed stain in pathology. This educational video aims to explore H&E staining, its components, and the characteristic staining pattern it imparts, making it an indispensable tool for pathologists and researchers. Hematoxylin and eosin, H&E, staining. H&E staining is a commonly used technique in pathology to visualize and differentiate various cellular components in tissue sections. This technique involves two main components, hematoxylin and eosin. Hematoxylin, derived from the heartwood of logwood trees, acts as a basic dye and selectively binds to acidic components in the tissue such as cell nuclei. As a basic dye, hematoxylin carries a positive charge, and it selectively binds to acidic components in the tissue, particularly to structures rich in nucleic acids, such as cell nuclei. Eosin, on the other hand, is an acidic dye and binds to basic or eosinophilic components such as cytoplasm. As an acidic dye, eosin carries a negative charge, and it tends to bind to positively charged or basic structures in the cells or tissues. In histological preparations, eosin is attracted to eosinophilic components, which typically include the cytoplasm of cells. This staining property helps to highlight cellular structures and differentiate them based on their affinity for eosin, allowing for better visualization and analysis of tissue morphology. The H&E staining process typically follows these steps. 1. Deparaffinization. 2. Rehydration. 3. Staining with hematoxylin. 4. Differentiation. 5. Bluing. 6. Counterstaining with eosin. 7. Dehydration and mounting. Characteristic staining pattern. The H&E stain imparts specific staining patterns to different cellular components, aiding pathologists in identifying and diagnosing various tissue types and pathological conditions. The characteristic staining patterns are as follows. 1. Nuclei. The nuclei of cells stain deep blue or purple due to the affinity of hematoxylin to chromatin, the genetic material present in the cell nucleus. This helps in identifying the number, size, shape, and distribution of nuclei, crucial for diagnosing malignancies, infections, and other pathological conditions. 2. Cytoplasm. The cytoplasm of cell stains various shades of pink due to the affinity of eosin to basic cellular components. The intensity of cytoplasmic staining depends on the abundance of proteins and organelles in the cell's cytoplasm, helping distinguish different tissue types. 3. Collagen fibers. Connective tissues, which contain collagen fibers, are stained pink by eosin. The presence and arrangement of collagen fibers are essential in diagnosing various connective tissue disorders. For extracellular matrix, components of the extracellular matrix, such as mucin or amyloid deposits, may also exhibit characteristic staining patterns under H&E, aiding in the identification of specific pathological conditions. 5. Specialized structures. In addition to the basic cell components, H&E staining can also reveal specialized structures, such as red blood cells, muscle fibers, and nerve cells, based on their distinct staining characteristics. Conclusion. H&E staining is a fundamental technique in pathology, allowing pathologists to visualize and analyze the microscopic structures of tissues. Hematoxylin and eosin, the key components of this staining technique, impart characteristic staining patterns to nuclei, cytoplasm, collagen fibers, and other cellular components, aiding in the diagnosis of various diseases and pathological conditions. As a cornerstone of histological analysis, H&E staining continues to be an indispensable tool in medical research, diagnosis, and education. Thank you for watching, and be sure to talk to your healthcare provider if you have any questions or concerns about your medications and any health conditions you may have. Disclaimer: the information presented in this video is for educational and informational purposes only. It is not intended to be a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified healthcare provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay in seeking it because of something you have heard or seen in this video. We would like to express our gratitude to our sponsor, First Cup Coffee, the original seed, for making this video possible. Shop firstcupcoffeeusa.com. First Cup Coffee, the original seed, is dedicated to providing high-quality coffee that fuels your studies and work with a rich and invigorating taste. We appreciate their support in bringing you valuable, free educational content. Remember to visit firstcupcoffeeusa.com for your daily dose of exceptional coffee. That's a wrap for today's lesson. I hope you found this video informative and helpful. If you have any questions or topics you'd like us to cover, let us know in the comments below. Don't forget to like and subscribe to our channel for more educational content. Take care, keep studying, and I'll catch you in the next video. Bye.